thank you for the introduction, and we're saving some time because the very first slide you've actually already seen. But I like pictures of sleeping babies because they always remind me that they're busy growing at that time. So today I'd like to speak to you about a different approach in terms of the kind of therapeutics that people would normally think about, certainly in previous decades. And so cord blood stem cells are in particular something that a lot of people have heard about, but often it's had more hype than real information. And there are many different kinds of stem cells, but the cord blood in particular is quite important. But really, in terms of, apart from what are they, what can they really do, and more importantly, are they actually safe to use in a way that nature didn't really intend? And I don't think I'm going to have much time to really touch on this, but other areas that we've been looking at, and I think Michael introduced, was the idea of protecting all organs during cardiac surgery. And so remote ischemic preconditioning is something we've been looking at. And what that is a process that really activates lots of natural metabolic type scenarios within cells that allow them to respond to injury, respond to stress, and this is in all tissues and organs of the body. And my other area of research, which is sort of related, really is the idea of the cardiomyopathies, and these are specific modifications within heart muscle cells that cause the heart to go into a failure. And in particular, we're also interested in not only the genetic sort that might affect things like mitochondria, which are important parts of muscle, but also sometimes interventions, like for instance, if a child is being treated for leukemia, the drug therapy may actually be, while toxic to cancer cells, may actually be toxic to heart muscle. And so we've done some work in that area and are continuing some major efforts there as well. But I'll only briefly cover that if I've got a bit of time. So the placental cord blood cells, well, this is something which we think we know. We think we know the placenta, and certainly this is what connects the child to the mother while it's developing in the uterus. The placenta actually looks like that, and this is really a remarkable organ, and really is what it's all about making sure that the factors that are delivered from the mother to the child that regulate gestation and growth and provide not only the hormone type signalling messages, but also the nutrition, but even more importantly oxygen. And even more important than that is the waste removal, and all of these things really are covered by this one organ, and it delivers it in a temporary input system that goes basically to the fetal circulation. And at birth, at the moment that birth occurs, that birth transition with the very first breath basically sets up a disconnection of this system, and it's quite a remarkable process and still not that well understood. Now, in terms of being able to deal with congenital heart disease in a way where cardiac surgery really redesigns the abnormally structured heart in a way that it can survive the first day of life, as Michael indicated in this morning's presentation, that cardiac surgery really has come a long way, and as we noted from the statistics, that really it's that last 15% that really that's what we're after in terms of what can we do to improve that. And so cord blood cells may well have an opportunity to work primarily in areas such as a hyperplastic left heart syndrome, but also other congenital abnormalities of the heart, in particular anything to do with the right ventricle or abnormal remaining right ventricle function, and other sort of anomalies and things like Epstein's and so on. But then there are other sorts of congenital defects that are probably not quite as severe, but can still take advantage of what cord blood cells might provide, so the transmission of the great arteries and so on. And then also a bit more complicated, but also cardiac myopathies are also, I guess, candidates. So you're probably quite familiar with the abnormalities within hyperplastic left heart syndrome. It's really probably the most severe of all of the abnormalities in terms of congenital heart disease. The main point is, apart from structural defects, is that there is a growing understanding of the genetic complications that underlie and what cause these defects to arise. There's not a single gene mutation, and in fact it's quite complex in terms of there being many different genes and very different, and that regulate very different processes. The majority of these, and I've listed some of these here, which really mean little except to say that they really are more involved with how the heart first starts to form. After first, basically it's about orchestrating all the different events and the timing of things to occur. And so because of that, we were really interested in the idea of, well, if these cells may be abnormal at that time point, when the heart's formed, later on, these genetic abnormalities actually don't have any real say in terms of what stem cells might be doing. And this is to just emphasize the difference between the hyperplastic left heart versus the normal heart. So we see this sort of very large muscular organ, although, you know, in a baby, this is actually very, very small, the size of the smallest of apples, I guess. If we see here, basically, the problem here is this left ventricle, which is basically absent. There's hardly any cavity. This is the left ventricle cavity here. And so clearly, it's a non-functioning left heart. And there are other abnormalities about the connections between the the right ventricle and the right atrium, valvular defects, but even more importantly is the fact that the aorta is very small. There are various connections that really are not going to allow the heart to pump. And remember, a pump literally needs good valves and it needs the strength to push blood through. And that's what the HLHS heart surgery really requires. And so there are three major procedures, as many of you will know. And in particular, the first stage that you must really provide within the first day or so of life is this Norwood procedure, and it really basically allows flow through the aorta, and so there's this enlargement, there's a grafting that goes on, and a number of different shunts and enlargements that are made to allow this procedure to occur. Now, surprisingly, first of all, the heart's exceptionally small to work on, so that's the first dilemma. Secondly, you require cardiopulmonary bypass to keep the body in good shape while the heart's actually arrested, so you can work on that. And then basically it's really the weight for the heart. Following the recovery of the bypass surgery, you've got to wait for the heart to get bigger, for the child to grow, to gain strength, and really then allow it to get to stage 2. And yet this is the most complicated, difficult time of all the stages, because if the right heart doesn't take over as the main dominant ventricle, then basically it will go into heart failure. And this remodeling process requires many things. First of all, it requires muscle growth. And we know that there are numerous complications to the cells in terms of, even though that they actually proliferate in the hyperplastic at much higher rates than in a normal heart, for some reason they're not getting all the messages that they need to boost the actual size and number of actual myocytes, the muscle cells that make up the right heart muscle over here. And so this is really the most dangerous time for the child, the first 3 months of life after the Norwood procedure. So we're very interested to try and boost the muscle and the function of that heart. So cord blood cells, while in utero, they play a very important role in terms of the formation of the body's cells, not just the heart, but all of the body's cells. And in fact, here we see a picture of the placental cord coming from the placenta with primitive stem cells, and there are many different kinds of these. And as far as therapeutics goes, at the moment, just like bone marrow stem cells have been used, these blood cells really have been used to treat mainly leukemic type cancers, and very, very successfully, mind you. And basically the collection is actually very quick and very simple. It's done aseptically, so as sterile as possible. And then there's a series of steps to process this, to extract out only the stem cell component. And then sampling is taken to make sure that there's no infection, there's no other complication, so that then you have a sterile preparation that can be delivered under clinical conditions. And it can be stored, and that's the other beauty of these. These can be put away and used later. So what do these stem cells do? Now we know that there are many kinds of stem cells. There are what they call a hematopoietic or blood type stem cells that give rise to red blood cells, white blood cells and platelets. But the mesenchymal stem cells, these are the stem cells that really give rise to the body parts that we have, from bones to skin, various organs like the pancreas, muscle, heart muscle and skeletal muscle, and the brain, of course, and many other organs like the lungs and the liver and so forth. And so because of that, the idea of regenerative medicine has been around for quite a while to use any kind of stem cell, but in particular any conveniently accessed stem cell might then be used to treat a condition that might cause a restoration of one of these particular types of tissues. Now, we've studied this and fortunately with the help of Heart Kids have supported preliminary projects to help us find out, well, what does cord blood actually have that may be of value. And we've found progenitor cells. These are cells that give rise to heart muscle. And we've been trying to work out what are the markers that tell us that this cell will become a heart cell. And in particular, there's this marker called IL-1 and Gator-4. We know that these give rise to heart muscle cells. And we've found that there are many, while there are many IL-1 positive cells, the ones that coincide with a second marker for the heart is actually even higher. It's like 78%. And so that's very, very important for us to know. But then we need to know whether they're metabolically active in a way that we know will definitely go towards being a heart muscle cell. However, those are about 1%. And you probably can't see that well in the figure there, but in this little area here, there's some green dots. And in those there, these are these cells that will become cardiac myocytes, and they work out to be about 1% of all these other dots that we see here. This is a measurement by flow cytometry where you can take cells and run them through a machine that basically counts them and identifies the chemical associations with those cells. Now we've tried to grow these cells together with actual heart muscle cells and try and stimulate to see whether one will tell the other, hey, why don't you become one of us and become a muscle cell as well. That's the principle. And we've looked for markers. These are chemical markers that tell us that these cells are becoming cardiac myocytes. And we can pick the ones that are human and we can grow them in a non-human cell setting so we can actually pick the difference so we know what the human cord blood cells will become. So it's just a way of marking these. And these can grow and up to about as early as 12 days in a dish. If my little film here will run, you might see right there where the pointer is, these cells are now muscle cells which are beating on their own, just as they would in the heart. And so this demonstrates for us that there is this capacity. But this is not all that these cells will do. And so this is what we're trying to work out, is that, well, they may well influence the formation of heart muscle, but there are other benefits which are probably even more important in terms of boosting heart muscle that's already in place in an organ. And within these cells we found the release of other messages, not just within the cells but also in the actual serum that the blood cells exist in. These chemicals called microRNAs, and these are very small nucleotides, for those who might know about nucleotides. And what's interesting about these is that they used to be thought of as junk. It was a message from the DNA that we didn't really know very much about a long time back. Now it turns out that these are actually extremely important in the fine-tuning and the fine regulation of what a gene does in terms of telling cells how to make proteins and what types of proteins to then build cells and structures and so forth. It's a way of getting cells to heal themselves by sending the right messages. And as we've found, that cord blood actually has at least 859 different microRNAs. And there are many different sorts according to different functions. Now, what's interesting is that no one particular microRNA is more abundant than another. In fact, the highest abundance of one single kind of microRNA is about 16%. And it turns out this particular message tells a cell about how to turn into a particular type of cell, whether it's a muscle cell or so on, how big to be, how fast to grow, how you do it, what kind of metabolism they should have, and so on. And as you see that the top 5 or 6 here literally govern most of the major processes that we need to see happen when it comes to providing messages to the body about how to grow. And so this is why cord blood delivered even after birth may actually be advantageous. And so this is something we're exploring further. In fact, one of the questions we've had, and we've been looking at this, is how different is this message profile that we have here, different from that we see in the hyperplasts. So far, we haven't been able to pick up any real difference, but we're exploring that with new tools to make sure that that's the case. In fact, we've been asking other questions about cord blood in terms of how different are the cord blood cells from the healthy kid cells, mainly because if a child can use their own cells, then this is really advantageous because they're already available, ready to go on day 1 or day 2 of life, and away you go, and there's no complications about where to get the cells and so forth. And in terms of what we've found is that overall the cells don't differ in number in terms of what they do, in terms of how they survive, and also what they become. And so although we're trying to build up our background data on this, so far we feel that this is really a viable proposition. So in fact, so much so that we've already jumped the gun and we've been trying to design experimental protocols that mimic what we see being performed in the operating theatre. And so we've used the neonatal lamb model, so newborn lambs within 5 days of life. But basically we have a surgical team doing the work just as would be done in the operating theatre. And so the Norwood procedure, the very first operation hyperplast baby will have, is basically like this as far as the time sequence goes. But during this time, we're preparing the heart for instrumentation. So there's a cartoon down here which shows, this is sort of cut open here so we can see inside where special catheters will go inside, and these will allow measures of pressure and blood flow and a number of different other measures, which basically is part of the instrumentation where we understand what's going on with the heart. And so what's happening is that when the heart's arrested, at the point here, beginning in red, there's a solution called a cardioplegia solution. It stops the heart. This is when bypass takes over. The bypass is keeping all the tissues healthy. The blood is still flowing through all of them. But in the heart's no longer pumping because the chemicals within the cardioplegia cause this heart to arrest. The surgeon can then operate and start to make all the sutures and various cuts and things that they have to do. Now, every so often, to keep the heart muscle alive, you have to flush fresh blood through. And what we've found is that at that time, it's very convenient to give, what we do is we take the dose of the cord blood stem cells and we infuse. You can see the dotted line here. And the last part of this reflushing, we give the stem cells, and we give it in part doses every 10 minutes. And then basically in the white part you see here, that's where anything that's not gone to where it should go gets flushed out. And so basically this approach which we've designed and the way that we deliver this to the blood vessels that feed the heart basically is very different to any other place in the world where the approach has been to actually just inject with a needle directly into heart muscle the stem cells. Now the problem with that is that two things. One, what you can get through a needle is very small and you can't put too many cells in a little hole that you make in the heart muscle. And then secondly, you're making a hole in the heart muscle, which doesn't really make sense. So by delivering through the blood vessels that pass through the heart muscle, we're actually sending the cells everywhere that blood should normally flow. And so it's actually a more direct and convenient way. So this experimental approach in animals has allowed us to be able to now show that the procedure, when done exactly as it would in the operating theatre, is basically uneventful, that it works just like a normal operation, and basically we don't take up any other or create any extra risk or create any other complication to the normal process. And the convenience of this operation also is that the time that the heart's arrested is only roughly around 40 minutes. And the operation is done in about 300 minutes altogether, and then all the other bits and pieces around that. But basically this is the most stressful time, and bypass is only about 100 minutes. And yet this creates a lot of systemic stress on the body. And so what we found that what you see here, this basically is a measure of the heart function, the way the heart beats. We see that on this side, this is a measure of the ventricular pressure on this axis here against volume. And so as the volume increases, here we see then the pressure goes up and so on. And so you can do this in steps, and the way these steps go up, this is telling us that the heart's able to create more pressure and pump more blood as the pressure goes through. After bypass, the heart's weakened, and we see that the slope of that line is less so. But this is actually a cord blood treated heart, and it's actually no different to the normal scenario. Now, what's interesting is that very few of these cells ever escape the heart. In fact, we can measure, there's only 3 examples where some cells got out, less than 1.1% actually got out. In the heart muscle, we don't see any of the blood vessels here, they're all open, there's no blockages, there's no blood clots. So basically, and also the cells look quite healthy, as you would expect to the degree of having had the stress of bypass surgery. Now, what's interesting here is where you can see these lit up, it's either green, red, or blue. These belong to the cord blood cells, and if we combine the image together, here we see that off the size of a blood vessel, we've got cord blood cells that have penetrated through into the heart muscle. And we can look at different regions of the heart, and different regions have a different profile in terms of where the blood cells go. Now, these are making their way from a blood vessel all the way into the heart muscle, as you can see there. And the great news is that 1 hour after the operation, all of these are there. What's interesting though was, while we didn't see much of these cells escaped around the body in the blood circulation, we did look into the various organs and did find a few blood cells here and there. Now, we didn't want them to go there, but the point is some did escape. But the point is also that if they're going to provide benefit, they will be providing it to organs that do suffer some injury and stress of the operation. And these organs here do succumb to the degree of stress from an operation. And so it's not surprisingly that these have gone to co-locate with cells in these different organs. Okay, so the bottom line here is that in terms of what we see after bypass surgery, that's the red. Basically, we see a decrease in function, and 1 hour after the bypass surgery, we don't really see, it's not worse, if anything, it's slightly better. Statistically, it's not significantly better, and again, this will depend on how many we show, but basically, the bottom line is that these are not creating any extra stress or injury. If anything, we feel it's going to be better in the long term. In the short term, we've demonstrated safety. And so now we're really preparing the human ethics, and we're looking to see what can be done to actually deliver and treat the first few children that come through. One last hurdle, of course, while we finally think we've convinced our ethics committees about the safety, we still need to set up a specialised laboratory that will take cells from the hospital at time of birth and allow us to actually have the preparation ready, but under the conditions that are required for certified clinical preparations. And I think I'm very close to the end of time, but just to give you an idea in terms of the other kinds of treatments we look at, here is a new protein called NDI1, which we're working on. This basically is a protein that mimics the missing protein that causes a lot of the mitochondrial cardiomyopathies. And just to show that when you can treat, this is treating an animal heart, what you see there is white in the red is basically injured heart. Okay, it's dying, it's what we call an infarct. And you can see that in these tissues, untreated, there's quite a lot of the white tissue, which means a lot of it's been injured and is dead. But in the top row, we've got a heart that's had this drug or this protein, and there's hardly any of it, even though it's had exactly the same amount of stress, the same amount of injury. And so this is one approach that we feel might be used to protect against genetic conditions where this is given very, very early on, and it will prevent, as we see up here, there is this enlargement that the cardiomyopathy causes. It can still pump, but it's not very functional. And the reason why it's not functional is, as you can see all this blue, that blue represents fibrosis. And so what it means is that as the muscle cells die off, the gaps are filled in with this blue fibrotic material. It's collagen and all sorts of things that try and hold what's left of the heart together. But it can't pump and it can't work very, very well. And so by giving this drug, not only does it prevent a very rapid injury, it can also prevent the long-term build-up of the fibrotic material. And finally, the other area we've been interested in for quite a while with our colleagues at the Cancer Institute at the Peter MacCallum is what happens to the heart if you've had a treatment for cancer. Many of these drugs like doxorubicin are very, very good at killing cancer cells. But what will happen in a child, they may survive their leukemia, but many years later, when they may be 9, 10 or even 14, so going into puberty, a time of great growth and stress on the heart, all of a sudden you have myopathies and a heart failure that can arise from that because heart cells were injured very early on, and then over time they actually get worse and worse. And so what we found, and I'll just show you the one slide which is very exciting, is this column here. The top shows heart muscle that's very healthy and aligned, very healthy mitochondria. These keep the muscle cells alive. This is a close-up showing how good they are. Basically, this drug, AN7, which is given together with the drug to treat cancer, basically it protected the heart muscle. And if you look at just giving the drug alone, you see all this disarray, this chaos, lots of holes, and lots of missing muscle. Basically, this is what the cancer drug does to the heart muscle. And so the point is, if we're able to design therapies that can protect the heart while you're trying to treat another condition, then this in itself will really be a major advantage. It will save a lot of grief a lot later, and certainly I think having what we call adjuncts to therapies really will make such a big difference in the long term. But our major focus, of course, is really what is done during bypass surgery to protect not just the heart, but all organs. And I'll just I'll eventually get to that. Here we go. So I'd like to thank Heart Kids, of course, for their funding support, and the surgeons like Christian Brizard, who's had this dream for quite a while to give cord blood cells to the Hyperplastic child, Michael Chung, of course, and Nari Elwood, who's the director of the Cord Blood Institute blood bank that's here on site, and the rest are fellows and members of my lab. So thanks for your attention.